So first I want to say thanks for giving me the opportunity to talk today here to everyone. You may have seen this story. It continues to develop, and we hope we're close to publication. As you can see here, it's a giant collaboration between a ton of labs and universities, and even across different 4DN teams. My name is Andrew Stevens. I've started at Drew as I go by more informally. My own lab at UMass Amherst, and I'm really interested in nuclear mechanics, as I'll tell you about today. But we're talking about how HP1 alpha is a chromatin crosslinker that controls mitotic chromosome mechanics. HP1 alpha is a chromatin binding protein here, seen binding as a homodimer to two different nucleosomes. Waiting for this to respond. It's not responding yet. There we go. So why should you care about nuclear mechanics and structure? Well, you should care because it's a hallmark of human disease. We have seen these changes in mechanics and structure across the human disease spectrum, as shown here on the left: aging, muscular dystrophy, heart disease, diabetes, atherosclerosis, and a large subset of cancers. We've known about this since we developed the microscope in the late 1800s. We started diagnostic tests in the early 1900s to look at size and shape of the nucleus in, say, cervical cells to diagnose cancer. That's called a pap smear. But more than 100 years later, we don't know what causes these abnormal shapes and what the real consequences are. And that's what my lab is very interested in. And I try to press upon you anytime I get a chance to talk to the 4DN, which is chromosome organization over space and time, is that you need to think about force as a fifth dimension that is going to influence these two things. And here, these disrupted shapes, I think, are a very clear way to see how the genome is directly impacted by force, by this, and by many other ways. So how do you measure force? I developed a novel nucleus isolation and force measurement procedure. Here we are isolating a single nucleus from a living cell with depolymerized actin so that we can get it away from the cytoskeleton. So we break open the plasma membrane with a little bit of detergent, and we can fish out the nucleus and suspend it between two pipettes. Once we have the pipettes set up, we can extend one and watch the deflection of the other. This is a force pipette. It's pre-measured for its bending constant. That means we get to back out its deflection by a pre-measured bending constant to get force. And overall, this gives us a force versus extension measurement. And that force versus extension measurement gives us lines like this, where the slope of the line is the spring constant. And what we know and what we were able to do for the first time was separate out the two major mechanical components of the nucleus, chromatin and lamins. And so I would argue that this tool is really helpful for the 4DN to separate out chromatin-based nuclear contributions from, say, lamin-based contributions. What we know is that chromatin dominates the short extension force response and is basically the base mechanical response of the entire nucleus. And that's up to about 30% deformation. 30% deformation occurs in most of your cells, less than 30%. Very few of your cells are going to see higher than 30% deformation. Those are those living in mechanically demanding environment or under migration. And in those cases, what you do have in those cells, you have increased levels of lamins to protect the nucleus, and lamins also are very important for determining migration. So here's the mechanical profile of the nucleus. And then how does that influence the shape? When we decompact the chromatin by increasing euchromatin or acetylated histones, which cause a more open state, we see that this mechanical profile gets weaker and the nucleus succumbs to cytoskeletal forces and deforms the nucleus. We know alternatively that histone methylation, and we say sometimes heterochromatin, is more important for the strengthening of the nucleus and maintaining shape. But we all know that heterochromatin is not only histone methylation, but also other proteins that interact with these histone methylations. Specifically, the one we're interested in asking about here is HP1 alpha. So can we separate out what the roles of methylated histones are from these other important chromatin binding proteins? So the Gruding lab at the Hutch built an amazing novel CRISPR-based HP1 alpha AID SFGFP line into the endogenous loci. And what the take home that I'm going to show you over 3 of these bullet points is that HP1 alpha is pretty dispensable for a lot of important heterochromatin functions. The first one you can see here is we can deplete the HP1 alpha in 4 to 6 hours, so very rapidly. And these local compaction sites that we see here that are enriched in both dense H2B and HP1 alpha remain upon depletion of HP1 alpha. So that means local compaction is not necessarily maintained by HP1 alpha. Transcription is another thing that's thought to be controlled by HP1 alpha. And in fact, we see no change in the transcription profile. I mean, there is some change, but this is, 40 genes out of greater than 16,000. So very little change. This is done for 4 hours. We also have it for 16 hours. So we have done this twice and essentially the same result. And finally, heterochromatin, we think of histone methylations. Specifically, HP1 alpha binds to H3K9 ditrimethyl, this constitutive heterochromatin marker. And in fact, over a 4-hour depletion of HP1 alpha, you get a 90% loss of HP1 alpha, but you see no change in the histone methylation, this H3K9 trimethyl. Okay, so if it's not important for many heterochromatin functions, what is it important for? And of course, I'm sure as you saw from the beginning of the talk, it's important for the mechanical response of the nucleus. And so here is our wild type control in black and our orange HP1 alpha degraded cell line. And we can see that it has a significantly decreased chromatin spring constant given by the slope for the chromatin-based regime. However, our lamin A-based strain stiffening, this difference between this dotted line and this increase here, is relatively maintained. So it's only changing chromatin-based nuclear mechanics. Here we can show you that this is true for the population. This is about a 40% decrease in chromatin-based nuclear rigidity. In past, when we have seen decreases in chromatin-based mechanics, we know that they associate with abnormal shape, and HP1 alpha loss is no different. We see about a doubling in the abnormal nuclear shape. Specifically, this is a measurement of solidity. We have also done this by nuclear curvature. Responding to reviewers' replies for our manuscript, we have now pretty much done this ad nauseum to find these effects that I'm going to show you. So the question is, how does HP1 alpha relate to histone methylation, these two things that make up heterochromatin overall? So we use this drug methylstat, which is a histone demethylase inhibitor. This will not remove methyl groups, so you'll have a bunch of methylated histones, which overall will cause you to have increased chromatin mechanics. We've also shown this ad nauseum in many other cell lines with many other different perturbations. And again, we see it here. So the question is, what is more important? HP1 alpha, does it determine all mechanical response, or does the histone methylation determine all mechanical response, or some in between where they both contribute? And the answer is it's the in between. So we can deplete HP1 alpha, get a weaker nucleus, and see abnormal shape. But then we can dial up histone methylation levels, rescue nuclear mechanics, and restore normal nuclear shape. And what this tells us is that HP1 alpha and histone methylation have separate mechanical roles. And so what could be that other role? Before we get there, I'm just going to quickly go back to why you should care, because mechanics and shape is a little abstract. You should care because weaker mechanics cause loss of morphology, which is associated with loss of compartmentalization. I apologize, this movie does not want to play. Let's see if we go back to, maybe it'll load better. And what you can see here is this is fluctuating, this is NOS-GFP, and it's going to rupture, boom, right there at the end. And so loss of mechanics means loss of morphology, means loss of compartmentalization, which is downstream going to disrupt nuclear functions. The nucleus is a compartment, and the loss of its compartmentalization will lose biochemical factors to the cytoplasm. You'll allow cytoplasmic factors into the nucleus, and this wreaks havoc. One specific example of this is DNA damage. And so we know that loss of compartmentalization, as well as a weaker nucleus that succumbs to deformations more often, can both cause increases in DNA damage. And in fact, upon HP1 alpha, we see a twofold increase in DNA damage. Getting back to function, what is HP1 alpha's job? If histone methylation per se is maintaining maybe polymer compaction state, what is HP1 alpha's job? Well, HP1 alpha is a dimer, and it could cross-link different things. Now, its cross-linking capacity is known to be both DNA to DNA, methylated histone to methylated histones, and there's also work done on phase separation, which we mostly think of just as a multivalent linker. So there's 3 different ways for it to act like a cross-linker. And all of those things are dependent on this dimerization domain. So what we did is depleted our endogenous CRISPR'd HP1 alpha loci. We get abnormal shape. We add back a rescue, an exogenous wild type HP1 alpha in cherry, and it restores shape. But when we add back a dimerization mutant, this I165E, that does not allow this cross-linking function or dimer function, we see that this does not rescue shape. So here we have normal shape, loss of shape, the rescue via wild type, but that is completely dependent on its ability to dimerize and cross-link different portions of the genome. Another way to look at this is just to look at it with essentially a cutoff for abnormal morphology we defined here. And it's very clear that dimerization is absolutely essential for its shape function, which also means it's likely important for its mechanical function, since mechanics maintain shape. So what is the model of how we think about this, right? So one of the things that the 4DN does is it takes a lot of experimental data and it builds simulation models. And so we've done that. The initial model was built by my colleague and good friend Ed Bannigan, who's now in the Mirny lab at MIT and also part of our center, the Mirny Decker 4DN Center. And what we know to get this two regime force response is there are two main mechanical components, lamins and chromatin, but we also needed to add chromatin-chromatin crosslinkers and chromatin-lamin linkers to get the proper behavior. So I'm going to stop there with the simulation and say, let's look at the experimental data first. Can we rule one of these out experimentally? And the answer is yes. Experimentally, HP1 alpha does not behave as a chromatin-lamin tether. Examples of chromatin-lamin tethers are PRR14, LBR, and maybe also LAP2 beta. They all have a peripheral localization that is enriched. So when we look at HP1 alpha's peripheral localization, we do a mask using lamin D1 to just grab the periphery. What we see is that HP1 alpha is not enriched at the periphery. Actually, it comes up with a 0.86 measurement, which is a little bit less than 1, not statistically significant, but it is not enriched at the periphery. And that is a behavior that is usually done in chromatin-lamin tethers. The other thing that these chromatin-lamin tethers do is they maintain heterochromatin at the periphery. So here's our H3K9 signal. And you can see that is it in fact enriched at the periphery. It's about a 1.5 fold partition at the periphery. That means it's about 50% more likely to be at the periphery than other places. And upon loss via HP1 alpha degradation by auxin, you can see that this does not significantly change your peripheral localization of H3K9 ditrimethyl remains. And so HP1 alpha does not exhibit the behaviors of a chromatin-lamin linker. So now let's go into the simulations and see what the simulations have to say. And the simulations are going to focus not on localization, but in fact on the mechanics, which is the model is built for. So simulations of nuclear mechanics recapitulate HP1 alpha as loss of a chromatin crosslinker. That's our take home. I'm going to walk you through how we get there. So in our model, we can titrate away chromatin-chromatin crosslinkers or chromatin-lamin linkers we call shell linkers. It was unclear that these were actually going to be different, but in the end, they were different, which is very cool that the simulation can provide us a difference that we might be able to see experimentally. So the chromatin-chromatin crosslinker decreased the chromatin-based nuclear force response, basically this portion of the curve, but the lamin-based strain stiffening remained the same. And if you look at these curves, they run very parallel to each other. Alternatively, the loss of the shell linker, or chromatin-lamin tether, resulted in a decrease in both chromatin and lamin-based strain stiffening. You can see here that this blue line and this red line have very different trajectories. That's likely because the forces are not being put off to the lamins. Remember, the lamins must bend first for the initial 30% strain and then begin to stretch later, which causes the strain stiffening effect. Experimentally, we observed the right, which is change only in the chromatin-based regime, and thus simulations suggest that HP1 alpha is best recapitulated as a chromatin-chromatin crosslinker, and this agrees experimentally with the idea that it does not behave positionally as a chromatin-lamin tether. We also looked at mitotic chromosome mechanics and function. I'm going to only go over a small portion of this. We really built out this portion of the paper in response to the reviewers. The one big thing we did find was that it's been reported that HP1 alpha is only at the pericentromere region. But if you think about how much HP1 alpha background is in the cell, it causes a lot of problems for seeing if low levels of HP1 alpha live on the arms. And in fact, when you isolate the chromosome bundle, the mitotic chromosome bundle, so this is the entire genome as a bundle isolated. This work is done by Ron Biggs at Northwestern. You can now see, once it's away from background, that there is a high increase, that there is a pericentromer foci, but there's also general staining across the chromosome arms. And we have a bunch of images of this. This is the one that I added here. When we ask if HP1 alpha contributes to chromosome mitotic chromosome mechanics, it does. What we find is that histone methylation tends to dominate. And if you go down here, I just want you to focus on this main point that there is a 40% decrease in the mitotic chromosome mechanics. So HP1 alpha has an interphase role and it has a mitosis role for mechanics. So it goes from about 260 piconewtons doubling force to about 150 piconewtons doubling force. Then we ask, does it provide a function? And in fact, it's also important for function. Let me be clear that we have not linked mechanics and function. We have seen them separately. I think it will take a sufficient amount of extra work to figure out if it's an HP1 biochemical or localization function relative to mechanics, but we see both of them and reported them in the paper. What we see is that upon loss of HP1 alpha, we see metaphase misalignment, and we see anaphase and telophase errors, mostly as anaphase bridges, and we see that this increases over the long as you deplete HP1 alpha for longer time intervals. So I'm going to stop there and sum this up, but then I'm going to have a little extra aside after this, which is that HP1 alpha is a heterochromatin crosslinker. So we think of histone methylation as a polymer compaction, and HP1 alpha is providing cross-linking capability. It provides stiffness from this cross-linking, and it maintains shape. And when it's lost, you lose shape, become softer, and you also have mitotic problems. I want to quickly parlay this into why this is cool. This is cool because at the same time, we were doing a different collaboration with the Decker lab, looking at how nuclear mechanics and high C provide evidence of chromatin-chromatin cross-linking. So they took the genome and they digested it infrequently or frequently. And what we found initially mechanics-wise was that when you digested it into 10 to 25 kb fragments, you would expect this polymer to have no mechanical strength. When we did the mechanics, we saw that it maintained strength. When we digested it further, we saw that it lost mechanics, as we had seen before. And other things, about 60% of its mechanics were gone. So what we think is, how do you maintain mechanics? You maintain mechanics by having a chromatin crosslinker to be able to provide the force to continue to move through the polymer, even when the polymer is digested infrequently. And with the Decker lab, which ran this entire project, it's an amazing paper. I was happy that they asked me to be a part of it. It shows that our mechanics measurements, which maintain upon infrequent digestion due to this believed chromatin crosslinking, also holds for 3D organization via high Z. So high C, even when you digest the chromatin to 10 to 25 kb fragments, it's still held together by what we believe are chromatin-chromatin crosslinkers. If you digest it a lot, then you lose that profile, and that can be seen here. Now we have evidence of how frequently the genome is cross-linked to maintain both its mechanics and its 3D organization. And the question is, who are the chromatin-chromatin based crosslinkers? I've told you here today that HP1 alpha, we believe, is one of them. Many of you are also working on proteins such as CTCF, cohesin, condensin, topoisomerase. There are a host of others that I either don't know about yet or that you will work on and you're going to tell me about later. Another one that's interesting is RNA. And so what I want to provide to everyone as a 4DN group is that we have a suite of tools to start to assess who are the chromatin-chromatin crosslinkers via mechanics and high C. And hopefully that's an appetizing sort of pitch for everyone. I want to finish by just letting you know that this work is on bioRxiv. We are working to get it accepted at eLife. We want to thank our funding in the 4DN. I do want to call out some people, which I didn't do a good job of calling out in the talk. Amy Strom is been sort of the data leader of this project. This collaboration, of course, is she's in Cliff Bringlin's lab. And so this shows that we are also collaborating across 4DN centers. She has really been the large data generator for the interphase nuclear biochemical measurements and a lot of great work. Ron did all of the mitotic measurements, and Ed did all of the simulations. And then of course, Joan and her team, the Green Lab, developed this construct. And so it's really been exciting being a part of this. And thank you for your time. I've just hit 20 minutes, so I'll stop talking now.